So today we have our last lecture, and it is not necessarily new information. It's kind of culminating with the cell signaling piece. I would have liked to have done this one first to introduce the whole concept of the nervous system, immune system, and endocrine system. But it was either a Wednesday and you were on your own, or it was a Thursday and you had a sub, I forget. And then the other one just seemed easier to give you. So we're going to do our cell signaling, which really is like all three of those systems together. We have a short practice sheet for understanding graphics. And then I have two CK12s in the Google world for you. And I'm not sure that we've done CK12s before. Have we? You might have done them in other classes because I introduced them to other people. Yeah, so it's based on the endocrine system. And then the rest of the hour, we have to just practice. I have practice books around the room. I'm going to print you the second practice test so that you can work on that. And you guys at home, there's lots of resources that I put in Google for you, or I'm sure you have your own practice books that you can use, or you have your own method. Yesterday, but I did the unit 4 yesterday. It was like the first minute and a half. If you do turn that on and it's a white screen, it was last year's Zoom and we were just learning everything. And it was actually before we were Zooming, but I forgot to share my screen. So it's a white screen for like a minute. But anyways, this one is on cell signaling and cell communication. So a lot of what I'm going to say today, I go into maybe even better detail in this review video. So if you want to look at that, you can. Hopefully you see cell communication, Anna Choi. Yes, okay, thank you. So cell communication, does this picture remind you of anything that we've talked about already? Yeah, good. So it reminds you of like helper T cells and antigen presenting cells, where the antigen is being presented, binds with the receptor on the helper T, decides if it belongs or doesn't belong, and then the helper T will release an interleukin or a cytokine depending on what response is needed. So that's a good example of cell communication. That is like a short communication, right? It's cell to cell communication. So that is what allows all of our cells to work in coordination, obviously. Unicellular organisms also do have a form of cell communication. It's called quorum sensing. Well, that's one example. Quorum sensing, so these are all bacteria and they're releasing these red markers. And the red markers would bind with receptors in another bacterium, which causes that, and they're not literally red markers, but in the graphic, and that would cause the next bacterium to also release that chemical. So this is a way for the bacteria to communicate and then cause a group response. So this might have to do with infection, where they would turn on some toxin response, or Bonnie Bassler, I talk about this in my video. If you get a chance to watch Bonnie Bassler, it's a TED Talk. She's one of the Princeton profs, and she works with this bacterium that lives in symbiosis with squid. And through quorum sensing, the bacterium sends signals to each other to tell when they get enough, a great enough density, they all turn on a fluorescing gene. And so they actually light up the squid. So that benefits the squid. Anyways, that she refers to it as like taking a vote. So when the density of the chemical or the signal is large enough, they're able to respond. Animals, we have what's called a gap junction. See the space between these two cells, so they might not be touching. We have interstitial fluid in between our cells, so there's a gap. So they're called gap junction. These proteins, these are like channel proteins. This is one cell, this is the other cell. So it allows two cells to transfer proteins, amino acids, different signaling molecules between two cells to allow those cells to coordinate. This is going to happen a lot in well, the heart, for example. The signal to contract happens in your pacemaker, right? The sinoatrial node. And it sends that, wrong side of my heart. It sends that signal to every muscle in your heart so that they all contract in unison. Well, not all of them at once. Like those are the atria, those are the ventricle. Anyways, so they are connected by these gap junctions, which send that impulse through the cells. Action potentials are sent through these gap junctions. And then also like during embryonic development, we need all of the cells to be changing morphine at the same time. So this allows for that communication between cells. So local signaling can be cell-to-cell recognition. You have local regulators. This is over a short distance rather than cell to cell. So we have surface molecules. This would be like our antigen presenting cells that we talked about, glycoproteins, which is what's on the surface of your blood cells, glycolipids. Then this is a local regulator. These cells can release other cells. And so then they become signaling cells. And this is common with growth factors. For example, this is called paracrine signaling. Para, remember, means around. So they're emitting signals from the one cell that gets the initial response or the initial signal. They're going to release chemicals that signal the other cells around them to all respond in the same way. So a good example of this is interferons. In the immune system, a viral infected cell will release an interferon, and it gets its name because it interferes with viral reproduction. So that interferon is released from the infected cell, and it's not protecting the infected cell, it's going to protect all the cells around it. So the interferon serves as a signal for the surrounding cells that there is a virus in the area. And so it turns on gene regulation, and they produce proteins that will then block the virus from gaining access to the DNA so that it cannot reproduce. So that's para, like around, paracrine signaling. And then we also have our neurotransmitters. So this is also a regulator. And we are familiar with this already because we talked about the nervous system. It works in that synaptic signaling. It has to jump over a gap. So these work only over a short distance, right? The neurotransmitter, if the gap was between your spinal cord and your toe, it's not going to communicate with your toe, right? It has to have a short gap. So that's called local signaling. So you have cell-cell communication, local signaling, and then long-distance signaling. So the endocrine system serves better for the long-distance signaling. We know that the hormones travel through the bloodstream, and they can go a long way, thus the name, to some other target organ or gland where they cause another response. In some cases, the nervous system can also participate in long distance signaling, but typically we would associate that with the short local signaling. We kind of don't think about plants a whole lot because we are animals, so it's easier to think about animals, but plants also have hormones that they release. And that would make sense. It has to grow and develop and change from a seed to a seedling, and from a seedling to a plant, and then from a plant to like producing flowers and fruit. So they have hormones that will regulate that morphism, if you will. And remember xylem and phloem, we talked about it during water potential. Xylem is the waterway and phloem is the food. Right, so the xylem, like where is our water in our bloodstream, right? 95% of our blood is water. So you can associate the xylem of the plant with the blood of an animal. So if hormones are traveling through our blood, then you would expect hormones to travel through the xylem, right? So it's going to be released, travels through the xylem, and then goes to, maybe it's to produce leaves, so it's going to go to the end of the stems. Maybe it's going to be to produce a flower. Maybe it's going to produce a toxin that keeps other organisms away. And then others are released into the air, like pheromones that might attract a pollinator. So no matter what type of signaling you're using, there's three basic pieces. You have the reception, the transduction, and the response. And now that we've talked about it, they probably already make sense to you. The reception, the target cell detects the signal molecule. So typically you're going to have a receptor protein on the surface of the cell. And I might just refer back to our protein structures. They often refer to the domain of the protein. So the transmembrane, the part that is within the membrane, they might refer to the transmembrane domain. And because this is inward for the protein, we would expect that to be nonpolar, correct? And then they might refer to surface proteins or the surface domain, and you would expect that portion of the protein to be hydrophilic. Totally side note, not related to cell signaling, but you see them talk about receptor proteins a lot in questions, and it's important to remember that piece because they'll talk about changing that receptor protein in some way. So you got a receptor protein that receives a signal. Remember the word ligand. So a signaling protein might bind with the receptor protein and then cause a response in the cell membrane. So we refer to that as a ligand. Transduction is that relay of information inside of the cell. So the conversion of the signal from this protein to something that the cell can recognize and respond to. So the final response, whatever the cell is supposed to do. Maybe it's supposed to reproduce, maybe it's supposed to produce interferon, maybe it's supposed to create another hormone. Okay, often we're creating enzymes or proteins that are going to be used by the cell or transported to be used by other cells. And this is, again, since we just did the hormone piece, it should look very much like a review. This is going to go into each individual step one slide at a time. So you have your signaling molecule, a ligand binding to the receptor. This is that lock and key concept. It's a protein. Anything that affects a protein will affect this receptor, right? pH, temperature. Why do you have to warm up before exercise? One, you're like, oh, so I don't tire a muscle, but also because your nerves are sending signals to your muscles, right? And if you want to have quick reflexes, for example, and quick muscle action, you're going to be crossing a gap, right? Acetylcholine is released from a neurotransmitter, crosses a synapse, binds with a receptor to cause a reaction in your muscles. The warmer you are, because molecules move more quickly, and we know that this will increase reaction rate, will cause that acetylcholine to move across the gap more quickly and cause a quicker response. Also explains why it's really hard to move your muscles when it's cold, because now that acetylcholine is moving very slowly and it's not crossing that gap very quickly, so it's not binding to the receptors. And then everything that's going on inside of your cell is affected by the temperature, right? The movement of molecules is affected by temperatures. And so that kind of explains why on a warm day, you're going to perform better, and on a cold day, you don't. Side note, but worth reviewing. So this ligand serves to cause a conformational change. Remember, we talked about allosteric interactions where it might bind and change the shape of a protein, and then the protein no longer fits its substrate. But in this case, it's changing it to cause a reaction. Puts it into an active form, basically. And the active form is part of this signal transduction pathway. This, you can refer to that secondary messenger, which is that G protein. So the signal is converted into a form that can produce a cellular response, and that is the signaling piece. So then the third is the response, whatever the cell needs to do. It might be catalysis, digestion, breakdown, right? It might be activation of a gene triggering apoptosis, programmed cell death, especially in cancer cells. It could be almost anything. So catalysis, breaking down molecules like glucagon, that is the hormone. Glycogen, breaking down glycogen into the individual glucose molecules would be an example of catalysis. And going back into these ion channels, notice the gate here. So these are gated ion channels. When the signal molecule binds, it's going to change its shape, change its conformation, right? So we can see this gate is now opening, which allows ions to change or to move through, sorry. So this is going to change the polarity of the cell because those ions bring with them a charge. So you call that depolarization. The two sides are becoming less different. Acetylcholine is a good example. We talk about illustrative examples. So if you understand one, you understand them all. Acetylcholine I use because I do anatomy and I'm already using acetylcholine. So that's a good one for me to use. Sarcolemma, anytime you see sarco, that's referring to muscle, and lemma is referring to membranes. So sarcolemma is the covering of the muscle cell. A neural lemma would be covering a neuron. So you can figure out words you don't know if you know the pieces and parts, right? Chunking. So a review of that cascade, because that is kind of confusing piece, right? So we had our receptor received a signal, activated a G protein, which is not necessarily shown in this, which activates CAMP, remember cyclic AMP, which is our primary kinase. So then that kinase, remember, they phosphorylate other molecules. And when they phosphorylate other molecules, they turn them into an active form. They have a conformational change, which causes them to activate. And so then it's a cascade of one phosphorylating another, kind of like sending a hot potato down a line, you know, that game as a kid, until eventually you reach your final destination, which is usually the nucleus and the DNA. This is the G protein portion of that. This side probably should have come first. So the G protein is activated by GTP, guanine triphosphate. And then that phosphate can activate the ATP conversion into CAMP. And then we talked about the other piece. The response can have sometimes different responses in different cells because it might activate different transcription factors. It can have the same effect in multiple cells. It can have no effect in one cell, but an effect in another cell. So this is just showing an example of a transcription response, which is a common response. So like that interferon might serve as a signal that turns on a transcription of a protein that will block RNA or DNA, block viruses from gaining access to your DNA, because that's what they need to do to make more of themselves. Speaking of viruses in DNA and RNA, remember retroviruses, they have RNA rather than DNA. So before they can transcribe, they have to first reverse transcribe into DNA. And then because we make proteins from RNA, they have to transcribe into RNA before making proteins. That was one of the questions from the practice test. So I thought I would throw that out there. So this is what I was just saying. This is one signal, two different cells. So we can see that it can have a different response in two different cells. Notice the end result here is different. It can also have more than one response in the same cell. So there's lots of variability. This would be a good example for like, how do we get so many different antibodies to fight off thousands of different infections? So we can have multiple responses within the same cell. It can act as an inhibitor or an activator. So I think we have a tendency to always think about a signal causing something to happen, but sometimes that signal causes something to stop, right? And then I feel like we talked about the other things, triggering multiple receptors. And then those different receptors would cause different responses because they're part of a different pathway. So depending on which pathway is triggered, you can do different things. All right, so kind of a review or a clarification or just a reminder of how the cell signaling works. A couple new words, the paracrine and the local signaling and the long distance signaling, but really we've already talked about the components within them. So what questions do you have? I have a quick practice for us in the Google Classroom. Oh, seven of you already did it. So I'm just gonna pick somebody's paper to pull up just for the sake of looking at it. So you often are given a graphic to use, some kind of long wordy paragraph to read. And then they say, oh, there's a malfunction in the protein, or something isn't happening, or you have too much of this or not enough of that. AP is notorious for changing the system. So this worksheet gives you, and I see that Grace is already working at it, so I'm not going to scroll it up. This gives you a chance to think about if you made a change to this system, what would the result be? So take a few minutes to fill this out. All right, let's see if you guys agree with these, because I didn't actually have an answer key, so I'm trying to make sense of it just like you. So if no insulin was produced by the pancreas, we wouldn't have a signal to take blood sugar out of the blood, right? So that would cause the levels to increase. If the GLUT4 protein does not reach the membrane, and the GLUT4 protein is supposed to shuttle glucose across, then blood will stay, or sugar will stay in the blood, and so it would increase. If insulin is being overproduced by the pancreas and insulin is taking sugar out of the blood, then it should cause a decrease in the blood. An antagonistic molecule, competitive inhibitor, binds to the alpha subunits and blocks binding. So again, if insulin can't bind, then there's no signal sent in to shuttle the glucose across. So it should increase. Mitochondria doesn't produce enough ATP. It's not really identifying where the ATP is being used, but I believe this transport protein requires ATP to function. And so that makes me think glucose would stay in the bloodstream because it doesn't have the energy to move to change this protein shape. And so it would cause an increase. Alpha helix beta sheet proteins have been denatured. So again, we need this shape to match the insulin. If it doesn't match, it won't do its job. So that will cause an increase. The GLUT4 protein, if it's denatured, once again, it won't be able to shuttle glucose across, so it should increase. And the number of GLUT4 transport proteins increases. So if I have more shuttles, I should be able to move more glucose. So that would cause a decrease in the blood. Agree with all those? Excelente. Okay, so this is where I want to go. There's 2 CK12s. You can do those now, or you can do those later. Practice, practice, practice. We have 45 minutes to just spend on topics that you feel you need to spend a little more time on. You can use any of those links in the Google World, or you can grab your practice book, start reviewing. In class, I have some of the practice tests that we did this weekend. They can't leave the room, but you are welcome to grab one and use that if you'd like. Helps you identify like what topic you need to study a little bit more.